So I'm going to talk about shapes and packing in biological systems, and I'm going to explain that using various Christmas imagery. So for the purposes of my talk, it would be really useful if everyone can imagine their body to be a very intricate, three-dimensional jigsaw puzzle, preferably a lot more complicated than this one that you can see of Santa. I don't know if anyone's ever seen that jigsaw puzzle that's covered in baked beans, and then on the other side it's covered in baked beans as well, and except that they're rotated 90 degrees. So that's the kind of hardness of jigsaw puzzle that we're talking about with the human body. There's billions of tiny little pieces, and they all have to fit together to form you lot, and they have to interact with each other. It's kind of like not a static jigsaw puzzle. So it's as if all the little pieces are constantly changing shape and then forcing the ones next to them to change shape. And it's so complicated that it's really impossible for one person to solve. I mean, completely impossible. And people in my field are sort of working on one very small part of that jigsaw puzzle with the idea that ultimately we can do the whole thing with lots and lots of different people's efforts. So, as I mentioned, because there are billions of pieces, they're much too small to visualize with your eyes, of course, but even with microscopes, even the most powerful of microscopes. So we have to use a series of kind of indirect methods to work out the precise shapes of proteins, or proteins are the main macromolecules that I look at, and so that's what I'll focus on today, but obviously there are lots of different kinds of molecules that make up the jigsaw puzzle of the human body. So unfortunately, because our talks are very short, I'm not going to go into any detail about my actual research and how we solve the shapes of these particular molecules, but I'm going to tell you a bit about how the body produces these molecules and how they get their original shapes that we have to try and then work out what they are. This is actually a slide from the seminar that I always give to research audiences, and I realized just by chance that it has a bit of a Christmassy flavor, so I've commandeered it for this talk. I should mention that one of the main reasons why we try to solve the shapes of all the jigsaw puzzle pieces is because when you understand the detailed molecular shape of a piece of machinery in the body, then first of all, you're in a very good position to work out what it does and how it does it. And also, if it goes wrong, you're in a strong position to design drugs that can precisely interact with its specific nooks and crannies, and thereby, any detailed shape information that you have it always comes in useful in one capacity or another. And in the spirit of interdisciplinary research, a lot of the techniques that we use to solve the shapes of proteins are coming from physics methods or chemistry methods to solve biological problems. So let me explain my snowman. I usually have to apologize because there are often some people in the audience who work on the real thing that I'm depicting here, which is called the ribosome, which is the factory that is present in all our cells that makes proteins. It does roughly look like that. This is a kind of low resolution image of it. So what happens is, as I'm sure all of you are aware, you have genes which contain all the instructions for making all the proteins that your body will ever need. And every cell has a copy of all those genes packaged up in a very neat way. So it's kind of keeping the instructions safe inside the nucleus of the cell. But when you need to make a particular protein, we have the mechanism to unwrap the particular bit of instructions, unwind them. I'm sure you guys know that DNA, which forms genes, is packaged up in a double helix shape, a fact that was discovered at King's, quite exciting. And so when you need to actually make a protein from a gene, you unwind that particular double helix, make a copy a kind of temporary copy, which is called a messenger RNA. And then you can use that temporary copy in the protein factory, the snowman, to make a protein. So what happens is the large subunit, or the body of the snowman, and the small subunit, or the head of the snowman, of the ribosome, they will converge, like they'll sort of come together around a messenger RNA. So the messenger RNA would be where you see the scarf, but then it uses the information in the message to start threading amino acids, which are the building blocks of proteins, onto a chain. So it's as if it has a string, and it's just adding different beads on as appropriate as it reads the message, which tells it which ones to add. And then it feeds the string of amino acids out from the snowman, and it starts to form elements of shapes, like I've shown it here, starting to form a bit of a helix. And the reason why I've got that number of shapes is because there are 20 different amino acid building blocks that go to make up proteins. The genetic information, as I'm sure many of you will know, is encoded in a similar way to how the binary computer code contains an awful lot of information using only a small number of, well, a very tiny number, 2, of different characters, 0 and 1. So in DNA, we actually have 4 letters, A, T, G, and C, which you might think might not be enough information to tell a whole body how to make all the different proteins that you need, and all the different shapes and all the crazy different mechanisms that are going on all the time to keep us healthy. I don't know how much audience participation I'm going to get, but can anyone think of any word that you could spell using just C, A, G, or T? I knew someone would say that. I was trying to be like QI and preempt what people will say. I picked Mog because it's the 90th birthday of Judith Kerr, the fabulous writer and illustrator of this book. So yes, as you can see from this, that you think there's only a limited amount of information that we can have in those 4 letters. But actually, there's a wealth of information, as you know, because I've already told you, that you can make all the proteins that the body needs. So I've created my own gene here. I actually just made it up for reasons which will become clear. I did search it in the database just to see whether it's a real gene, and it actually is a real gene. It's part of a much larger protein that's found in mice. It wasn't exactly present in human, but something quite similar. Anyway, so the way that you get the coding for 20 different amino acids from only 4 letters, A, T, G, and C, is that you have 3 letters of DNA code that codes for 1 amino acid. So if you have 3 letters of the genetic code, you can actually have 64 different combinations of 3. And as I mentioned, there are only 20 amino acids. That means you can have what's called redundancy. So it means that several of these little combinations of 3 can encode the same amino acid. And this fact, actually, it comes in handy for lots of different processes, including evolution and including a lot of the techniques that we do ourselves in the lab to study genes and proteins. Again, I don't have any time to go into that, but I'm just going to use this to demonstrate how the cell will make a particular protein. So the letters AGC, there's a little code where you can look up which codon, as it's called, stands for which amino acid. And AGC is a serine, and CTC is a leucine. And before I carry on, I just wanted to tell you, in case anyone doesn't know, that all the amino acids can be represented in various different ways. Obviously, they have a defined chemical structure, as I'll show you in a moment, but they also have long names, so examples serine or leucine. Then they have a 3-letter code, which is usually the first 3 letters of the long name, but sometimes it's not, but mostly it is. So for serine, it would be SER. For leucine, it would be LEU. But then they also have a single letter code, as I'm showing here, the S and the L, which is useful for my Christmassy purposes, as I'm sure that you can start to guess what I'm about to do here. I just wanted to mention that some of the amino acids start with the same letter, so they can't have the same single letter code. So for example, alanine and arginine, they both start with A, but alanine is called A, and arginine they use R, and that's quite easy to remember because it starts arginine. And there are some others like that, like phenylalanine starts with P. Proline also starts with P. So the proline gets the P, and the phenylalanine gets the F because it sounds like it starts with an F. So anyway, some of them are not so intuitive, and that's what I was getting at before I show you the next one, because GAG is actually encodes a glutamic acid, where there's not very an intuitive way of why glutamic acid should be encoded by E, but you can make weird ways in your head to remember it. And then ATC is isoleucine, GGC is glycine, and CAT is histidine. And I'm just going to show you the chemical structures of these amino acids as well. The thing you'll see that they have in common is they all have this alpha carbon at the bottom, and every alpha carbon is bound to 4 things. And the 4 things are always 1 hydrogen, 1 carboxyl group, and 1 amino group, which is why they're called amino acids. And then the final space is filled with something different each time, and that's what gives the amino acids their unique properties. So some of them are big, some of them are small, some are positively charged, some are negatively charged, and some are hydrophobic. They like to be in a kind of a sort of fatty, oily environment, and some prefer an aqueous environment like the cytoplasm. And all these different properties threaded along the primary sequence or the string of a protein make them be able to fold up into their specific shapes, which are really important for them to do their jobs. So I changed it into some Christmassy shapes, just for my purposes of my demonstration. And as you can see, although they look faintly familiar, they don't really look like anything useful when they're just lined up in a straight line like beads on a string. You actually need them to all fold into their correct orientations in order to become an actual machine that can do something. So a protein is no use, and if you think about all machines, it's very rare to find a part that's just a straight line. You need it to form a 3-dimensional and useful mechanical entity like this. As you can tell, this image is probably missing some quite crucial things. So sometimes when you've got a protein that's folded into its correct shape, it still can't necessarily do its specific job because it might need some accessories or helpers or direction for where it ought to go. Can anyone think of anything particular that's missing from this image? Yeah, and various other things. So, so with a bit of Christmas magic, I can give it all the accessories that it needs, and I can let it go off into the sunset to do its job and deliver all the presents. Okay, so just wanted to say thank you.